Hi, everyone. Welcome to another webinar brought to you by the C45 Quality Association. For those of you that don't know by now, my name is Tommy Leonelski. I'm the chair and the host and moderator, chair of the association, and also the host and moderator of this webinar. Before we begin, we're going to let people log in, get settled for a few minutes, and I'm going to do a little spiel here about our association as well as our upcoming summit. So I hope you enjoy this webinar, and we're really looking forward to kicking it off. So the C45 Quality Association is quite a unique industry association in Canada. We're a non-for-profit organization, and we're membership-backed, not corporate-backed, like some of the other industry associations. We were started about 4 years ago now, I believe, by industry professionals who thought that the quality, regulatory, and lab testing folks that work in this industry needed a unified voice. So currently, we represent about 100 individuals across Canada, employed in these quality, regulatory, and testing kind of professions for various license holders across Canada. And really, we just strive to be this unified voice for all these people working in the industry, and we do a number of things to advance this mission. So first and foremost, we put on public and members-only virtual events such as these, typically on a monthly basis. This allows us to kind of network and just have a little bit of a community and discuss the most pressing issues in our industry as they relate to quality and regulations. We also have in the past hosted in-person events like our Quality Summit, and we are actually bringing that back this June. So I'll let you know more about that in a second. And I think one of the most important things that we do is we regularly interface with various regulators, especially Health Canada and the provinces. And we like to distill the issues raised by our members and the industry at large and provide constructive feedback on a regular basis to those organizations. And we do that on a regular basis, typically quarterly. And it's really great that they want to listen, and our relationships with those organizations have been great thus far. If you want to learn more, check out our Twitter, YouTube, or LinkedIn channels for latest events and news. Sign up on our mailing list at c45association.com to kind of be in the know about any upcoming events. And really, please consider becoming a member. Memberships start at $49 per year for students, $99 for non-voting. And really just helps us run this association. We're a team of 7. No one on the board is paid. It's all volunteer based, and we just have general expenses like accounting and legal fees and website fees. And so your memberships help with that, as well as planning in-person events. So please check us out if you want to learn more, and we're looking forward to having you as a member. Now, I did want to mention that we're bringing back our C45 Quality Summit. We have planned it for Friday, June 7th, 2024 in Hamilton, Ontario. We want to gather about 200 people, folks from government, both federal and provincial, industries such as labs and processors, and even academia, and put them into a room together to kind of, discuss and do what we're kind of doing today, but an in-person event. So it's a one-day event, fully catered. We're going to have panels. We're going to have presentations. We've invited government and industry folks to kind of present and attend. Should be a lot of fun. So please consider coming out. You can check out c45summit.com to buy tickets. Tickets are available for $99 right now for members. It's early bird until March 31st. Tickets are selling quite fast, so please make sure to get one because prices will go up and almost double as we get closer to the event. So again, the more support we have early on, the better for us to plan this event. And we're also looking for speakers, ideas, and sponsors. So if you're a lab, if you're a license holder, if you're a software provider, and you want to have the ability to interface with some of the most important people in the industry, please email us at theteam@c45association.com. Thanks. Okay, so I know we all want to get into this webinar. There's well over 100 of you on here. This is really great. Just a bit of housekeeping. This webinar is being recorded, and it will be available on our YouTube channel. Please subscribe to that. It should be up there in the next few days, and there's an archive of all of our past events as well. We've left plenty of time for Q&A after this presentation. We're hoping to have at least 20-30 minutes of Q&A. So please use the Q&A feature in your Zoom client and post all your comments and questions. The panelists and myself will be kind of monitoring that box. And just please keep your comments positive and respectful. And if there's any technical issues, let us know in the chat. We'll figure that out right away. And yeah, it should be a lot of fun. So without further ado, I want to kind of introduce this topic and some of our presenters. So what we're going to be discussing today is one of the most important topics today when it comes to quality assurance and lab testing, and that's this problem of potency inflation. This is an issue that we at the association have raised for many years, and many others have as well. And it's not just a Canadian problem. We see this in the US and other legal jurisdictions. And, for better or for worse, THC content is one of the most important variables for people when they make a decision when purchasing cannabis, especially dried flower. But right now, there are very few controls in the current system to prevent inflation. Samples are collected by license holders. They could bias the samples. There's rumors of certain labs reporting favorable results. The test methods themselves are not inspected by Health Canada, and provinces demand high potency products. In fact, a lot of their websites even have filters for high THC products. So all of this contributes to a lot of finger pointing, a lot of confusion and distrust in the industry and consumers. And of course, there's international implications as well because we export cannabis around the world. So what we're going to do is hear from a number of analytical chemists today that took their matter into their own hands, decided to see whether potency inflation in the Canadian market is an actual thing, and whether labs here in Canada can report similar results, even if they use different equipment and methods. So I'd like to have all of my guests turn on their microphone and camera and say hi to everyone. And we'll start with Rob. Rob was the kind of leader of this study. So I'd like Rob to introduce himself, and we'll go from there. Thanks, everyone.
Yeah, fantastic. I'm really grateful for the opportunity. I'm a former university prof that's been involved in the cannabis industry since legalization. I've been involved at all levels from QAP to executive to et cetera, setting licenses. And I've gotten all types of licenses, from hemp licenses to testing licenses, processing and cultivation licenses. So I have a good breadth of experience there. But I've also been actively engaged with the natural health product sector, and I'm currently with one of those companies. And I've also been involved with AOAC and ASTM, both of which are very active developing standards in this area. Oh, and by the way, one of our presenters, Denise Johnson, who was very important to our workflow, was going to be presenting, but something came up last minute, so she won't be here.
Fantastic. Thanks for that, Rob. Next, I'd like to introduce Hubert Marco. Hubert is a colleague, director of the C45 Quality Association, but also one of the founders of Laboratory Phytochemia. Go ahead, Hubert.
Thank you, Tom. So I'm Hubert Marceau, and I'm one of the co-founders of Laboratory Phytochemia. We are a lab who specialize in natural and plant phytochemistry in general, and we've been working into the cannabis industry since 2014, so just before the legalization. We have been involved into the C45 Quality Association in some way. We have been involved internationally in some association into the natural product sphere, and we have been also pretty well outspoken about this situation, which we have been observing since 2018.
Fantastic. Thank you, Hubert, for participating. Next, we have Thomas here from Variant Labs. Thomas, please introduce yourself.
Hi there, my name is Thomas Fraley. I'm the founder of Variant Laboratories. We're in Mississauga, Ontario, just by Pearson Airport. We're a GMP and ISO 17025 accredited lab. We mostly focus on cannabis. We do domestic testing for Canadian cannabis, as well as GMP testing for cannabis bound for foreign export markets as well too, with a little bit of work on the side in natural health products, dietary supplements, and pharmaceuticals.
Thanks, Thomas, my old stomping ground in Mississauga. And last but certainly not least, we have Andrian Rakov. Andrian is from RPC and has been a guest on some of our webinars over the last few years. So Andrian, please introduce yourself.
Thank you, Tom. I am currently the Director of Research and Controlled Substances at RPC. We're a research and technology organization that does a lot of analytical chemistry testing and specialized engineering and research work. We got involved in testing cannabis actually back in the late 90s. through hemp testing. We were one of the first in the country to have a hemp testing license. And from there, we transitioned into testing medical cannabis and have been in the space quite a while. And we're very excited to be part of this interlaboratory study and a group of very much like-minded, analytical chemists. And we're trying to demonstrate that, or share our perspective of how we kind of fit into the whole cannabis industry, and hoping to dispel some myths with some of the results that we present today.
That's great. Thank you, Adrian. Okay, everyone. I think we're ready to start, so we can throw the presentation on the screen here. And Rob, I believe the floor is yours. Good luck, and we'll see you on the other side. And just a reminder to everyone throughout the presentation, please use the Q&A box, put your questions in there, and we'll get to them in kind of priority sequence after the presentation. Thank you.
Yeah, like I said, it's my pleasure to be participating in this and presenting this data to your audience. I think it's very important to do this, if for anything else, because of the just the context of this. Can I get the next slide, please? Okay, so there's a lot of questions when we started this, is why this is important. There was a belief that the THC values on packaged materials were elevated, and there was a lot of people pointing fingers and arguing with different types of problems. But rather than do that, take that approach, I guess our thought was, let's look at what's there, be transparent about what we find. We've never really outed anybody that was causing a problem, but when we find things that were outrageously out of spec, we did report to Health Canada, and hoping that their internal mechanism would improve that. There's an issue of consumer trust and confidence with the legal supply, but also about 20% of the people that are purchasing from the recreational market are buying it for medical issues or almost like over-the-counter medications. But really, most importantly, the Canadian regulatory framework is really the first of the G20 to legalize. So it's being used as a template for other areas looking for legalization. And this is a great example for us to lead the world by showing how this is done properly. Unfortunately, because of concerns with the public, THC or CBD content are primarily focused on the label claim. And because of that, it became a primary quality indicator. And it appears that many consumers are buying cannabis based on the THC content. So having elevated values there's a marketing advantage. So it's almost a fraudulent thing if they're putting artificially high values on this. And so this led to a lack of trust. But really, what got me is that internationally, when we were exporting materials, the line came out that Canadian labs cannot be trusted for this international trade. And the results were unreliable. So that this impacts not only the cannabis sector, but also the natural health product sector, and potentially the pharmaceutical sector, because some of the same labs are involved in testing these. And I believe in order to solve a problem, you have to properly identify it. And to identify, you have to look honestly at what's there, and honest and transparent testing is the way to find solutions. Can we get the next slide, please? So, before we even jump into this, I want to take a second just to talk about variance in a measurement. We really never know what the true value is. If we test frequently enough, the average is an indication of what the average value is. But another important metric is the variance within the product. And I think part of the problem with the cannabis is that individual buds have a significant variance. And if you do one single COA on a harvest, the odds of that being representative of what are in a package is very low. And when you do a test, each step in the process can add variance. So you have variance from the population itself. And a good analogy of this, if you just weighed a whole bunch of people, you wouldn't expect them all to have the same weight. And the average weight, when you calculate that, you wouldn't expect some small set to have exactly the same weights, right? So, and we wouldn't start questioning the ability of a weigh scale to weigh people. But the method variance, yes, there is. When a single lab is doing testing over and over, there'll be a variance even if they're testing the exact thing. So that's sort of in-lab variance. When two different labs test and they have subtle differences in their methods, you could have between lab variance. Within the packages, if you test a series of 5 different packages or 3 different packages, there is between package variance that will add in. And all these variances add together. It can be indicated by the standard deviation. And the square of the standard deviation is the variance. So that's another complication that comes into play here. And in some cases, people have suggested that the labs cannot test because they sent a series of samples to different labs and get different results back. Some of that is normal, but also some of the labs could be leaning into giving higher values because they get more business that way. Or the person sending it out could be sending out biased samples, and you can't distinguish those two things. The next slide, please. So where we started with this, I initially took it on myself to test some products in the marketplace in BC. And I looked at a variety of products from 7 grams and 3 grams and 1 gram. And I think we tested almost 50 products with that. I tested multiple packages on these, and we found that a significant amount were significantly below label claim. The question, of course, is, was my lab able to do accurate testing? So with that, I took, when we took the package, what process is we homogenize all the material. I only need about 0.2 grams to do the test. So then I had residual material, and I sent it to 2 different labs, and the average results for those were within 0.2 of the values that I found, 0.2 milligrams per gram. So that was an indication that my results were consistent with what other labs will have, but that was us homogenizing it first, and so they weren't involved in the sample prep. So then what we wanted to do is to involve more labs so that we could really understand the variance between labs, as well as the argument that labs without the same method aren't able to do accurate testing. So what we did is we took some homogenized PT grade material or proficiency testing material. So PT Canada does proficiency testing in Canada. And what they do is they send out vials that have powdered homogenized material of 2 different types to different labs, and we get a consensus value. It just so happens that my lab was involved in preparing some of those materials for a recent study. And we had some residual. So in order to, if you like, qualify the labs that were involved in this work, we sent them 2 different materials to each of the labs. But we also sent them some pre-rolls as a real sample. And we bought 3 packs of 20 pre-rolls. And from this, we took 2 from each of the 3 different packs. So each lab got 6 different pre-rolls. And we then were able to assess whether the labs were consistent with the PT material, as well as how they were consistent with the pre-roll material. What we found was that the relative standard deviation was about 6.5% between the labs and the measured data for the pre-rolls. What this is important is that PT Canada publishes the results from PT studies from a variety of different areas, including environmental samples. And our results were consistent with what were found for similar analytes from the cannabis sector, as well as for things like phenolics in water. So the cannabis labs are not performing any differently than environmental labs, and there's no suggestion that the environmental labs are not able to test materials that they're analyzing. So in order to show that, can we go to the next slide? What we did is those 6 different pre-rolls to 8 different labs is 48 different samples. We let the labs use their own method that they were using, and also to provide insight on how to do the sampling. And in one case, one of the labs left some of the paper material from the pre-rolls in with the sample, and there's a variance there. So they had a little bit higher variance in the other labs, and that kind of skewed the distribution. But to show the distribution, I've simply plotted all the data points on a theoretical Gaussian distribution, and you can see that they're distributed quite well. One of the values that were measured was outside of the range, and that is has what's called a Z-score of greater than 2. And Z-score is just the multiples of the standard deviation. But the other 47 are plotted on the curve here. And for comparison, we also have the label claim. So you can see what a group of testing labs would get on that material. And this was 26% below label claim. Clearly, the label claim was not reflective on this from the measured values. Health Canada does specify that for finished products, testing has to be done in the finished product. And I think that if it was done, you clearly wouldn't get this result. My speculation, and it's just a speculation, is that the company who produced these products used the harvest COA to put on their product rather than testing the finished product. And if they did with any of these other 8 labs, they would have had somewhere within that voucher, which they didn't. We reported lots of data to Health Canada, and unfortunately for dried flower and pre-rolls, there is no enforced variability limit. Although there is terminology that suggests that differences of 25% per unit, but I thought that would meant THC content, but it didn't. Health Canada's interpreted that just to the mass of the product or other things. But anyway, that's what we've done. So with this, we've kind of qualified the lab so we know that they can produce similar and consistent data. And then we also refined the method that we're using for pre-rolls, so that you'll hear about with the next speakers. And now I'm going to hand it off.
Thank you, Rob. The first part of the study was to establish if the group of labs that were selected were reliable, meaning that we didn't add any specific or major skews from each lab method to ensure that if we were working on the same material, we will produce the same result, which was quite well demonstrated because we had pretty tight RSD. The second part of the study was once we had the certainty regarding the lab capacity to do the testing, we wanted to test the material directly bought on the market. However, we could not really buy the same material and send it to all 8 labs because they're usually on the market. You'll find small 3.5 gram sample. So the way that we work for that is that we bought flower sample from the market and we distributed amongst lab in a semi-random pattern. So each lab would test, each group of 3 lab would all test at least the same product once. And we split those against 3 different categories, meaning that we had dried flower product, we had 4 pre-roll product, and we had infused pre-roll product, each of them having their own particularity. The dried flower being a raw material was expected to have higher variance being the raw plant. The pre-roll product was expected to have lower variance since they are already milled, already processed. And the infused pre-roll, it was expected to be a little bit more complex process because each infusion process will be slightly different. So some would have keef, some would have crystal and stuff like that. We'll touch that a little bit more that later. And they were bought directly in store, and they were sent to Rob Lab, where they were dispatched in a semi-random pattern. All in all, that's for 142 different products that were tested over 34 different brands in 3 categories. Each lab received 6 dried flower products, 6 infused flower products, and 3 pre-rolled products in their original package, meaning that there was no subsampling from after the sample acquisition. So each lab would have received exactly the same product as if they would have bought them into a local store. And lab were also told, such as in phase 1, to use their own internal method, but we did give some specific direction on some of the product. So for the test methodology of dried flower, each lab use, most lab use 0.2 gram of dried flower after homogenization and extracted with 20 milliliter solvent. Some differed a little bit in the ratio and the mass taken, but all in all, each lab had a ratio of about 1.1 gram per 10 milliliters of solvent. There was also different solvent usage. Some lab were using 100% methanol. Some were using a mix of 80% methanol and 20% water, such as is suggested in the AOAC method. And some lab would be using the 90% methanol and 10% chloroform extraction solvent. And each lab had their own HPLC, different HPLC, somewhere UHPLC, somewhere HPLC, each has their own elution solvent, gradient, detection method. So we didn't add any direction in this regard. We wanted that to be as close as possible as to what each lab would be doing for any customer. So for the results, we tested 16 brands. You can see that each product was tested by at least 3 labs. In some specific case, there were 4 labs. This is just a random thing. There was no intention behind it. During the sample selection, we did add a slight skew regarding higher level of THC. So you will notice that most of the label claim are around the 30%, with the few that are under it. We also tested during the study, we tested for CBD, total CBD, so the acid and the non-acid form. We also tested for CBN. However, due to a low level of reporting and concentration into the material, we decided to not use these value during the study. So in this table there, you will see that the first column is the sample ID, which is a random ID. The other one is the lab use. Each lab were assigned a letter. The total THC that were measured is the average of each measurement, and the RSD is calculated based on the standard deviation. It's a relative standard deviation, meaning that we expect, for example, in the first row, we expect the total THC measure to be 190.9 milligram per gram, plus or minus 6.3% of 190.9 milligram per gram. For those who prefer to have the value in percent, you just have to move the period to the left. So for example, 190.9 milligram per gram is 19.09%. The label claim is what was found on the label on the product that was bought in the market, and the percentage of deviation is how far the value that was measured is from the label claim in relative percentage. So for example, in the first row, the percentage of deviation is minus 41.8%, so over 40% deviation. Afterward, the 2 other columns are the 95 confidence interval range. These are representative of, if we would say that each lab, as we said previously, is not measuring the true value. It's always an average. We never knew in healthcare chemistry what is the true value of a product. So if we measure, if a lab measure something, it is expected that the real value is somewhere in between a certain range, more or less close. So the way that we do that is we assume, due to the Gaussian distribution, that the 2 times the relative standard deviation, or the standard deviation, will give us a 95% confidence interval, meaning that we are not sure at 95% that these values are equivalent to that the value that we measure is 95% certain to be I'm sorry, that the true value is 95% certain to be in the range that we have set. So for example, for the first sample, based on the RSD measure across the tree lab, we expect that the real value to be between 167 milligram per gram and 215 milligram per gram. It also means that, for example, if the label value would be in this range, then we would just consider that the label value is a good one, because there are still 95% chance that the label value is the same value as the one that is measured. We have added a color system to show different categories. For example, for all samples that are more than 40% over the limit, they are coded in red. All samples that are more than 30% over the label, they are coded in light red / orange. And those who are over 25% are in light green, and those who are within the 25% variation limit, which is arbitrary, but that we selected to use, are colored in green. I also bold those whose label value are included into the 95% confidence interval range. The result of that is, for the dried flower, is that over 16 different brands, and in the 53 different packages tested, 9 brands were more than 25% below the label, meaning that, this is including of all the other range. 6 were more than 30% over below the label, and 2 brands were more than 40% below the label. So that leaves us with only 44%, so roughly half of the sample that we tested, which for which we would consider the labeling to be correct. However, one very important distinction here is that if you are taking a random assortment of reading, we would expect some to be over the reported value and some being under the reported value. In the case of dried flower, no value were over the reported value, meaning that there were in any case there was an over representation of the high value, which is quite a normal if you look at it onto an analytical point of view. It is also important to note that Health Canada has no variance limit on dried flower, which give no incentive to ensure that the label value is within a certain tolerance, such as the 95% confidence interval or something that is similar. So from there, I'll leave the podium to my next to the next speaker, which would be Thomas Fraley from Variant.
Thanks, Hubert. While Hubert was presenting, Rob did notice a typo on the slide for the flower data. Yeah, if you could go back. So I actually just quickly edited this now because it's a live document that we're sharing. So initially this had been presented with a decimal place here before the last digit. This had been misrepresented. It was actually the weight percentage value that got put in here instead of the mg per g. So I just quickly went and corrected this all to be the mg per g value, just so there wasn't any confusion. So now you can see here where the delta is coming between what the label claim was on the product and the actual measured value here, because initially you would have been seeing something like 32.8, which wouldn't have made a lot of sense. So we just quickly edited that here to highlight that. So thank you, Rob, for noticing that as we were going. It definitely makes a big difference in the data we're presenting. So yeah, so coming on to my little segment now here, I'm going to talk about the 2 other products that were tested in the study. So Hubert just finished testing kind of like the dried flower. And now I'm going to go and talk about our data for the regular pre-rolls or non-infused pre-rolls, as well as infused pre-rolls here. So again, just emphasizing here that all labs used our own in-house validated methods. Generally speaking, for a non-infused pre-roll, and this will become relevant when I talk about infused pre-rolls after, our methodologies are all very similar in that we will typically take the cone and empty out the contents and then directly analyze the contents. So the paper is not typically included in the analysis for infused pre-rolls. It is a small amount that gets added in, but it's important to consider in the context that we're looking at here. Very similar to how dried flower is analyzed on its own, because it is just dried flower. Typically, most labs are using about 10 milligrams of extraction solvent per 0.1 gram or 100 milligrams of test material. Similar solvents were used in this because, again, it is still just dried flower like before. So a variance of solvents. We use 100% methanol here. Other labs use 80-20 methanol water. Some use 90-10 methanol with chloroform. Again, we all use a different range of instruments for conducting this test as well too. So next slide, please. And I think we're correct here on our mg per g units. So I just wanted to check that out first. So this is correct on this slide here. So similar to the dried flower results, you'll see of the 4 different products that we tested, we did have one that came back substantially different. We had about a 50% difference in the label claim. So this label claim is 297 milligrams per gram, which would be 29.7% weight total potential THC. I did see another question that came in from someone, which I answered with text, but just wanted to clarify as well too, that when we talk about label claim for THC here, yes, we are talking about total potential THC or total THC. That is the sum of delta 9 THC plus 0.877 times the THC acid concentration to account for the decarboxylation when you're smoking. So again, some of these were close to the 30 or 25% category here. But this is the data that we got on the non-infused pre-rolls here. So we can go to the next slide, please. This is, again, just representing that same data. We did have one that was quite a big outlier here. That was around a 50% difference. And then the remainder here were within the 25% bracket, although granted that one was quite close to the 25% that we saw as well, too. Notably as well, too, Health Canada has no variance limit on pre-roll, much like dried flower. We did make the note here that generally it is in the guidelines that products should be tested in their finished form. We point that out just to account for the fact that sometimes when you're processing the flower from, say, its bud form to its milled form, and then transferring that milled material into a pre-roll, trichomes are sticky, they stick to stuff. So again, we can only speculate as to where the variance in these values come from, but one possibility is if you're testing at the bulk phase before something gets subsequently handled and put into the pre-roll, there's a possibility that you're going to lose trichomes in that process and that your end product could have a decreased potency. Again, just speculating on potential areas where these deviations could be coming from, so we wanted to draw attention to that. Can I have the next slide, please? Okay, I'm very excited to be the one that gets to talk about this chunk of data here. I think this is very interesting. So again, infused pre-rolls, just so we're all on the same page here, a variety of different forms these can come in. I tease my team by calling these the nightmare samples internally, just because as an analytical chemist, it's interesting when your target is both inside of the pre-roll and also stuck to the paper as well too. So you can see here a couple different examples. It might be hard to see on your screen, but an infused pre-roll can be something as simple as milled flour that has THC acid diamonds blended into the inside, in which case it's sort of similar in consistency to a standard pre-roll in terms of the powdery content. But you get really messy ones sometimes where you have the paper itself or the cone itself is actually dipped or rolled in something sticky like distillate, and then perhaps even rolled into keef afterwards as well too. So generally for these samples, what the labs do is we do include the paper in the analysis, because there's no way for us to quantitatively scrape off all of the analyte from both the inside and what's infused into the paper. So again, the labs all have their own methods. Internally, we would typically cut off the entire cone past the filter, and then we cryogenically mill with liquid nitrogen here, and then subsample our portion from that for analysis. Each lab might have their own workflow, but generally we're taking the paper as well too in this case, because it has our target inside of it. Similar extraction solvents as the previous data set, different instruments used as well too. Next slide, please. Okay, this is what I find fascinating. Look at the color, just look at the colors alone here in terms of delta between the label claim and what we measured. So it's very interesting to see, we're all sitting back and talking about the challenges that we're facing, with some of the discrepancies between label claims for dried flower and pre-rolls and maybe what retest values are getting. We got generally speaking, very tight retest values when it came to the infused pre-rolled products here. Again, we're still at the speculation point about where this could be coming from. If one was to look at it from a cynical perspective, you could say you're already adding in distillate or concentrate. Why is there any pressure to bias or inflate the results anymore? Another way of looking at it as well too is because these are classified as concentrate products, there's more regulatory pressure on the acceptable deviation for the results versus the label claim. So I thought this data set was really, really interesting to look at how, generally speaking, for these infused pre-roll products, we're seeing much closer retest values versus label claim compared to the dried flower in bulk form as well as pre-rolls. Next slide, please. Again, this is just a representation of that data as well, too. You'll see we don't have any of these getting into that red category that we identified. This is just summary here. And again, a note that because these are a concentrate class product, there is this 15% variance limit to account for as well, too, which might put more regulatory pressure on the on those results and their, I guess, retestability. So that concludes my slides. I think the next speaker is Andrian.
Thank you, Thomas. So I'm going to be wrapping things up before we segue into some discussions and Q&A. So I think some really interesting findings, and overall, you know, we saw that many dried flower products were outside of you know, that 25% window in terms of the label claim, and in all cases, as has been mentioned a couple of times, always consistently below. We didn't see a single example for the flower products of something being above label claim, and again, we would expect that if, you know, if things had kind of a natural variance, that, you know, some would be a little bit above label claim, some would be a little bit below. But in this case, we certainly didn't see that. So things are definitely seem to be biased in one direction. Also, as Thomas highlighted, interesting with the infused pre-roll category, where, you know, as soon as there was a regulatory variance limit in place, most of the products that we looked at met that requirement. And one of the things I was, you know, excited to kind of see between, you know, what was the variability between different testing labs using their own methods, you know, different people testing in what we started out with being fairly consistent products on different days, you know, and having all of that kind of variance in place. We saw that, you know, labs that were using their own validated methods, and if we were testing material that was quite homogeneous and similar, we were getting really similar results. And I think that speaks to, you know, the ability of laboratories to be, you know, both repeatable and reproducible. And sometimes, you know, we hear a lot around, hey, things could or potentially be better if everybody was using the exact same method. I think here with, you know, 8 different labs all being able to, you know, albeit within a slightly more controlled experiment, being able to demonstrate that the variance between the labs is actually quite low. You know, we can definitely, you know, produce results that are comparable. And you know, we're excited to kind of continue to demonstrate that again in the future, based on some of the things that we saw in this study. Certainly, I think there's an opportunity to try and take a look at, you know, putting in place some variance limits for dried flower and pre-roll products that currently is not in place. And it may be the case that is what is necessary in order to get everybody on board and say, hey, look, you know, right now there's no real restrictions on how close you need to be to the label claim, but this could be a great starting point to try and tighten things up a little bit and give people a better idea of, you know, what the actual true total THC concentration in the cannabis products are. So none of this would have been possible without all of the various labs that participated, and we have them all listed here: BCIT, Pura, RPC, Northern Scientific, Laboratoire Phytochimia, Supra R&D, Organogram, Vivariant Laboratories. And I'd love to highlight that a lot of the work was really, you know, in-kind We donated our time and materials to making this happen. And we were really grateful for some financial support from the C45 Quality Association and also from one of our participating laboratories, Organogram, that helped provide some of the monies to offset the cost of buying all of the products that we bought, the 150 some odd samples to distribute around. So, this was certainly done as kind of a labor of love, if you will, to try and get all of this data together and make this happen. And it was very much, people coming together, good sense of community, and with a kind of a clear objective in mind, and with very little, I guess, financial incentive to do so. So thank you to everybody that has contributed their time and resources to this. Much appreciated. And one of our participating laboratories as well had funding through NSERC. So we'd also like to acknowledge their contributions to making some of this happen. And I think we can get all of the other panelists, Thomas, Hubert, and Rob, if you'd like to pop back up, we can start launching into discussions, and I think there's already a bunch of questions that have been popping up.
Absolutely. Adrian, on behalf of the association and all of those in attendance, as well as Hubert, Rob, Thomas, and I see Denise is attempting to join us as well. So hello, Denise. Thank you so much. I think this is very enlightening. We at the C45 Association, we're very happy to provide some funding to offset some of these costs, because let's be honest, this is expensive kind of work, and this is where we like to put our money where our mouth is. We take the resources from our memberships and put them towards projects like this, and we hope to do many more in the future. So yes, now we have about at least 20-30 minutes. We'd love to have a lively, long discussion with well over 150 individuals on this call right now, many from Health Canada, from the provinces, from the industry. Let's get to the questions, and just as a reminder, this is being recorded. So if you have to leave early, you'll see it all on YouTube very shortly. I do have one or two questions I'd like to start with before we kind of delve into the Q&A here, just because as a kind of lifelong skeptic, I love busting myths. And some of the discourse I've heard over the years was the fact that, labs, you can't trust labs. They're all random number generators, and you all run labs, work in labs, or professional analytical chemists. Does anyone want to comment on the results of this study specifically, but also just generally your relations with other labs, and kind of push back on that? Because to me, it's quite clear that even if you're using different equipment, you have different analytical chemists conducting the sample prep and the analysis and different methods, you can still get pretty darn close inter-lab results. So I just wanted to kind of kick that off and hopefully put that myth to bed. Does anyone want to kind of take that on?
I'm happy to if nobody else wants to jump in.
Please go ahead.
And I will apologize. My joke is I'm a reformed academic, so I can go down rabbit holes. I guess the truth is that if a lab properly validates a method, and two different labs have properly validated methods, they should be producing similar results. That's how it works in every other sector, from environmental to healthcare to everything, right? So the key thing is properly validating the result. The reason why it's useful to include ISO 17025 accredited labs is that they have experts that look in on their validation records to ensure that they've done it properly. But the reality is, if you properly validate your test methods, in some cases, like these were inter-lab studies as well, that not only are they validated within one lab, they're validated across multiple labs. You do not need to mandate exactly what that is, as they did in California. They mandated what the method is because they were trying to solve what they thought was the wrong problem. And the big exception that I have is that the method that they're mandating is inferior in many ways to the method that we're running in terms of runtime and other quality metrics. So of course they can. We know that. We've accepted that across the board with other sectors. There's no difference in finding environmental contaminants versus or natural health products, botanical actives. We've been testing these for, yes, there's variance, but we can understand those.
Thanks, Rob. And I think that brings up a good point. You know, we've had a recent webinar about the benefits of accreditation, third-party accreditation, like through CALA or ISO 17025. As a former QAP in the industry, when I would hire a lab to do my testing, one of the first questions any QAP should ask is, "Hey, can you show me your validated methods?" But you know, I'd get a 1 or 2 page document with someone's signature on it, and really, I'd have no insight into whether that was actually a good validation, unless I really push hard. And some labs are just not happy in sharing that. So I guess the question is, we know that there's a lot of labs that do ISO 17025 accreditation, but what role does Health Canada, the federal regulator, play or has played? In other words, maybe this is a open question to anyone. In the last 1 or 2 years, how many of you have had an inspector from Health Canada show up to look at your cannabinoid potency method or the data your lab has generated and perform some sort of critical analysis on that, and just to determine that what you're doing is accurate? Can anyone speak to that, please?
Yeah, I think I'll jump in. I also want to go back a little bit on your random number generator. And when we're validating a method, we're especially trying to establish what is the variance of the method. We're always assuming some kind of random variation into the method, but we expect these variation to be in between a certain range that we deem acceptable for the purpose of the method. And this is a very important thing, is that for each method, a method has a purpose. So it must be, so the need of the industry, the need of a customer needs to be taken into account during the validation. And regarding the Health Canada inspection, I would say that from what we've heard, we have been starting to see some inspection going around, and quite frankly, we will have one in 2 weeks, and we are pretty happy to that, actually. You especially when you are working into an accredited environment, you learn to love these audits because they are always good opportunity to improve and to find maybe flaw that you didn't see or that you didn't expect to be flaw. So having a third party look on your work is very interesting, and it's very it's good for us, basically. And what did in the legislation, Health Canada asked for the method to be validated based on the ACH Q2B guideline, which are pretty clear and that require a few different parameters to be validated. And while it's easy to say, okay, this parameter is checked, this parameter is checked, this parameter is checked, all these data need to be available. They need to be available to auditor, and for us, we also made them available to customers. So if you see your validation method, I don't know for each of us, I will only be speaking for us, but you have all our data. So you can see what kind of variation we have, at which level we have them, and for what are each parameter of the method. And it's pretty transparent, to be honest. And it need to be transparent. It need to be, it need to, we need to communicate what are the difficulties of a method. So I saw that one of the question here is, why is 25% being suggested as the allowable variant for dried flower? Actually, it's the variance that is suggested. I think it's 20% by the USP, because they know that there's a lot of variance into this product. Some products don't have that much variance. If you look at metal alloy, for example, or for liquid, we don't expect such variance. But in these natural products, it's not expected. And all these large variance that is expected, and that is well defined when we're doing validation in each lab, may look to some as a random number variation, but in fact they are not. It's just that if you look at a very small subset of data, you end up with a larger you have a higher chance to just pick up number that are more spaced apart. But if you start bringing them all together, such as what we did with this study, where we have 142 data point, which is starting to be a considerable amount of data point, then you see trend appearing. You see that no, they're not random. They follow a trend. They follow a different. They are within a certain range, and this is something that the industry need to take into account. When I was showing the 95% confidence interval, one point that is important to me is that if your confidence interval is between 19 and 23, then if I say 19 and 23, for me, they are the same number. They have the same value. So if you are expecting to have a 0.001 variation, all lab know that this is not possible and that this is not true, and the validation showed it. But you need to be conscious as a QAP, and you need to be conscious as an industry that these variations are not sign of something going wrong. They are a sign of something going well, in fact.
So if you don't mind, I just want to get back to some of the points that you made in your question.
Yeah.
And there's a couple layers there. So first of all, you said as a QAP, you sent out to different labs. The reality is that it is hard to, because within a plant, there can be 10 or 15% variance within a plant. And within a harvest, there can be 20, 25%. It's a characteristic of the skill set and the growing conditions, et cetera, right? And then if you add on top of this, the processing, which, as Thomas mentioned, once you lose trichomes, the numbers change. So two things happen once the variance increases and the average value comes down. When we do PT studies properly, we use powdered homogenized material that we do everything we can to make sure it's the same material to compare labs. You can't just really do that with just dried flower, so it's hard. The second is having QAPs be the one responsible for determining if the labs have properly validated data. Let's just context this. They don't necessarily have the background, but when we do an ISO 17025 accreditation, there are experts with more than 20, 25 years experience that come on site that spend 3 full days to make sure that our stuff is properly validated. Now, not all the labs in this group have received their ISO 17025 accreditation, and from the data they've presented in our group, they're perfectly fine. But there's very few QAPs that have the skill set to assess the ability of the labs to do this. So from day one, I've encouraged Health Canada to either require 17025 or put some kind of proficiency testing as a condition. The only thing they seem to care about is that I don't have criminals in my lab, and I make sure that I have control over the material. They've never once that, and I've gotten probably about half a dozen different licenses for that. They've never once asked, can you test the product? Yeah. And so that needs to be part of it, I think.
Yeah, and therein lies the problem. And I think us having these conversations, the industry, our association, other associations, you yourself doing these studies, I think we're starting to see the tides turn. And just like Hubert mentioned, I have heard rumblings that, you know, Health Canada has been knocking on laboratory doors and specifically targeting some of this. But again, the regulations as they stand put a lot of pressure on the QAP and the lab themselves. And there's just this kind of there's a gap there, and you know, I think we all hope that that someone else like an expert like an ISO accreditor could fill that gap if it's not Health Canada, in order to give some confidence.
Yeah, just on that point, I'll also, sorry. So if a producer sent me material that was either doctored or very premium flower, and I reported the results for 35% THC, and that went on the label, I would have done that completely ethically, but I would have been providing a high value. If I'm not sent a representative sample, then I can't be responsible. The other thing is, when I receive a sample, I homogenize the whole material. I don't cherry pick. I don't de-stem. That's what I believe you have to do to do this properly. And I only report on that value. If they ask me to look at the package value, which is what we've done here, you can see the labs can get very close to a consensus of what that is. And it's not what's on the label currently.
Rob, and that brings me to my final follow-up, and I think you're reading my notes here before we get into the Q&A. I wanted to, bust another myth here because people often point to labs that, the labs are misreporting, but like you just said, you're at the whim of the sample that you're sent. So I'm going to pose this question to everyone on here. In your career or careers, has anyone here seen evidence of salting or purposely biasing samples? We know that kind of happens in mining and other industries, but have you ever received dried flower where it's not supposed to be an infused extracts and you found extra keef, THCA isolate, distillate sprayed on it? Have you ever gotten a flower and said, how come this is testing at 35% when I know that anything over 25 is essentially getting to the upper limit potential of the plant? So I'll kind of turn it on to everyone, and then we can start going through this list because I know there's a lot of questions, but we do have another 15, 20 minutes here at least to talk. So does anyone want to take that on?
I'll jump in here and just kind of share, our kind of our testing history and perspective. In the years that we've been involved in cannabis testing, the number of times that we've received a flower product that has tested at or above 30%, I can count on one hand. It's very rare for us to actually get a sample that is in that range, and certainly because for us it is not the norm. We always go back and retest those just to make sure that it wasn't something in terms of our sample processing that could have produced those results, just because for us it's not the norm to see values that high based on our historical data. But I don't think we've seen samples that would have been contaminated and would have tested really high as a result of that.
Yeah, and
A little bit similar
it's not
here.
part of our question, right? So we're asked to, you send me a package, I test the package, right? But I think your mention of 25% is incorrect. When preparing PT samples, we have bought kilogram quantities that we've used to make that. And we have one supplier in BC. I was actually amazed that it was over 30%, but it was consistently throughout the batch. They were just exceptional producers. So we can't rule that out. As we get things going and the demand for THC goes up, there'll be higher values there. So you can't just draw the line. So I have seen that, but it's not the question you're asking. And the thing is, we charge $80 for potency testing. I'm not going to prostitute myself for $80. And the thing is that the idea of focusing on lab shotting and it's labs, it's like blaming the prostitute for prostitution. There are people that they'll bend things to help you. But the people that are making the ones that are putting 40% on their package and selling all the product out and having the distributors want to buy as much as they'll produce, right? They're the ones that need to be accountable, as well as the people that are buying that, the distributors. They need to, if there's a problem there, they need to own it. So that's my thoughts.
Internally, when we see a more than 30% sample, we usually double check our data. We don't really redo the analysis, but we at least ensure that the data are consistent, because it's still possible that we have a flower with 30%, and that we are a sample with 30%. However, they are uncommon enough that they do raise little flags. I also agree with Rob here, is that as these industry mature, we will tend to have higher push toward higher number, but there are some number that are still preposterous. And also to confirm what Rob is saying is that we need to analyze what we receive. I mean, if you send us a bud in a kilo of hay, then we would just homogenize everything, and then you will have a very low level. While if you send us the top bud, then we don't have the power as a lab, because we don't always know why the customer is doing the analysis. Maybe you want to test this top bud, maybe you want to test the whole lot. And we don't have to report anything to Health Canada, and we don't have the tool nor the authority to ask our customer, did you do the right sampling? Is this sample for labeling purposes? All that we can do is test the best that we can, everything that we receive in the best practice possible and return to the best of knowledge what is the best result that we can.
I'd like to
Thanks.
chime in a little
Yeah.
bit before we move on, if that's okay. So two
Please.
things to point out. One, I know we use 25% as the variance in this as kind of like the green zone. Just something to consider from a thought standpoint is that in a lot of these export markets like Germany and things these days, they're using 10% variation as an acceptance limit. So whether or not that's going to be reasonable in practice or will be changed is something that will be certainly very interesting to keep an eye on in terms of acceptable variation from the label claim. Another thing too is that like I often get told in advance of these things I have to play nice and I'm not supposed to say names and stuff like that. But I also want to be very careful of sitting as a lab panelist in front of industry without sort of saying that there are no bad actors in the lab space. I think Rob mentioned in the beginning, the truth is that there is financial pressure to deliver high THC results. And I do think that we can have bad actors on both sides of the equation and that that's a reality. And so this is truly where I do see more regulatory I would say participation as being very critical to help stamp that out. Because truthfully, if a bad actor lab has a bad culture of quality and corruption, it doesn't matter how many certificates or what they do or how many accreditations they have. There are still ways of putting your thumb on the scale to tip the values. And so this is where I see small developments like the Ontario Cannabis Store, OCS, starting to retest on stuff that comes in as a really positive development as well too. I wish we would see more regulatory participation with retesting things as well too. Because again, I want to be very careful with sitting here. I do believe that on both sides, both at the producer level and the lab level, there can be influence, but I definitely don't want to point the finger at one versus the other here as well too. I also just want to point out as well too that regardless of what happens from the regulators going forward, if we look at the US jurisdictions as any kind of like prophecy of things that could happen. Another thing that you might start to see is attention from law firms who are going to start to realize that simply from a consumer standpoint, selling a consumer something that's mislabeled and not what they're actually getting could be grounds for a class action lawsuit as well too. So this is very important for people to consider going forward as well. Even if we don't have regulatory intervention, I think eventually we're going to start to see legal participation on this side of the coin as well too. And so I don't want to be too salty or point names or be Mr. Negative, but I also want to be very careful about sitting here and saying that there aren't any bad actors on the lab side in the market as well too. And I just felt that was important to say.
Thomas, that's incredibly well said. Thank you for stating that. I think we're all kind of thinking that, and I think it's important that our audience knows that as well. Thank you.
So just to follow up on one of the things that Thomas said, imagine a scenario that if products were on the market that were more than 20 or 25% out, if there were consequences, if led to a recall, and if the cannabis wholesalers also had to pay this hefty fine, then the onus would be on finding the labs that give you the best, most accurate results. Right now, if they give you high results and you can justify putting that, the only consequence is you win, and nobody does anything about it. If we change that just subtly, things will improve. In terms of the 25%, one of the reasons that I focused on that is there's a per unit descriptor that says 25% variance. So I said, okay, let's go with that. But there's also things in the regulations that say if you're doing a stability test and values fall outside of 20%, then you have a product that fails stability. So all these products that are more than 20% out are effectively failed stability for shelf life. So that's another way to get to that, right? But there's lots of ways. And this evidence is like when Health Canada set a limit for 15%, which is hard, particularly for heterogeneous infused pre-rolls, almost everybody's in line, and they're able to get it. When they don't have a limit, like with dried flower, almost everybody's out of the box, right? So why wouldn't they? Why wouldn't you? That's the problem.
What a coincidence, eh, Rob?
Just to add one more thing,
go ahead.
Just looking again at our dried flower data we presented, out of most of them were all over 30% label claim. Only one of them actually tested over 30%. So it's not that it's impossible, but certainly not impossible, but it's not the most common value that you would typically see, at least at this stage from our results.
Another interesting point regarding the data is that in the infused pre-roll, it was the only data set where we had measures that were over the label, which is indicating of a good representation, meaning that these were probably properly measured and sampled.
Yeah, I
Yeah.
saw a question come in on the Q&A about that as well too, about how when you look at all in a truly like randomly distributed data set, you'd expect that if things were measured properly, our retest values would come in both slightly above and slightly below. But in the case of dried flower and non-infused pre-rolls, all of our 100% of our values are all below the label claim. And so that is statistically relevant.
Absolutely. That's a great point. Okay, gentlemen, there are 24 questions here as expected. So what I'd like to do is actually give you some of the power to pick and choose some of the most important ones. So if everyone can start looking at the list and kind of choose one or two that they want to tackle, I'll give you guys a minute to do that, and then whoever wants to go ahead. But before we do, there is a first question here from Jacob who asks, will this presentation be distributed after the meeting somewhere? And I know we're recording this. This will be on YouTube. But will this PDF of this presentation be available, or are there any plans to actually publish this in a peer-reviewed publication, perhaps?
Yeah, can I answer the peer review? Yes, this material, we've done a lot of work with the previous. We are looking at a couple of publications on this. One just on the data itself, but the other one on how to properly approach an in-store study with an interlab group. I think we need guidance on people, how they do the testing, so it's fair to the industry and that's taken care of. So yes, there will be a couple publications, and we're currently working on that.
Fantastic. And is anyone open to sharing the PDF version of this presentation as is, or yeah? Okay, we'll have that posted on our a link to that on an email blast to all the registrants after, or on our website. Thank you for that. Okay, let's kind of turn it over to all our participants. I'd say we have another 10 minutes, so let's start with one question each. Whoever wants to go ahead, please just go ahead and rephrase or read the question and then answer it, and then anyone can also follow up, okay?
I've seen a few questions regarding the package date and the COA and the shelf stability of the flower. I briefly mentioned it, but in the data set that we acquired, we also test for CBN, which is usually the indicator of cannabinoid degradation. It was not significant at all. We decided to not include it. There were no flower with the significant CBN level into any other product. I just also want to reiterate that the sample selection, we always took the same lot. So if we had 3 package, we made sure that the 3 package were at the same lot number. I don't think we did any filtering for the package date, but I do believe that we have this information into the data set. However, as far as I know, it didn't brought any significant difference into the results.
Yeah, I'll add to that. And from even some other studies that we've previously done, separate from this study, we looked at packaging dates, year, 2 or more, and looked at if there were any significant changes in the cannabinoids, and again, nothing that would kind of explain any of the discrepancies. So I don't know if it's certainly something you have to consider, and in some cases, depending on how it might have been stored or whatever else, we could have led to significant degradation, but nothing that we saw in this study that would account for those large discrepancies we observed.
Does anyone else want to tackle a question in the Q&A?
There is a question regarding the solvent system for extraction efficiency. The 3 solvent system that were used by the lab, the 100% methanol one, I think is the most common. I also think that it's the one from the European Pharmacopoeia. I think that the German DAB is using 95% ethanol. The 80% methanol, 20% water is actually the AOEC reference method solvent, and the 19 methanol, 10% chloroform solvent system is the one that was published from the De Baker et al. Method that was kind of the grandfather of all the method that are currently used, and it was also used by the Dutch government and Bedrocan back in the days. Most of us did some solvent validation during their method validation or some testing regarding the solvent system. Most of us, if not all of us, all came to the conclusion that the solvent system, at least the most common solvent system, don't have a large impact on the value. We do, however, based on this study, have a follow-up question regarding the official AOAC method solvent system that may give slightly lower results than the other solvent system, but this is still an hypothesis, and we didn't verify it during the study.
Yeah, just to speak to solvents and validation in general. So whatever solvent that you use to do your extraction, one of a good practice is certainly to do what we kind of call exhaustive extractions. You do one extraction with the solvent, you kind of pour it all off. Then you add a fresh amount of solvent to whatever you're extracting, re-extract, pour it off, and analyze those portions separately and see, okay, did you extract 98% of your target analyte in that first shot, and you're only missing 2% in that next extraction, or is it more pronounced than that? So that's usually something that labs will look at when they're trying to validate. The European EP method, I think that Hubert mentioned, they use 3 subsequent extractions with ethanol and then pool them all together. I think every lab has a different workflow, and as long as you've demonstrated that whatever solvent you're using and whatever process you're using, and I think we've done that in this study. We've shown very equivalent results despite some of the differences.
Thank you, Adrian, for that. Okay, I would say another 5 minutes if we have the time. Does anyone else see any questions that they want to tackle? If not, I might have 1 or 2 for the group before we sign off.
Yeah, I'll just mention, so somebody was asking about, why do we think was the root cause of some of this stuff, particularly with that. So, and I do have some theories of what it might be. In a given harvest, as I said, there's an average value and a variance between different buds. We never measure the variance, but those are 2 characteristics of that harvest. But really the thing is, if I use a harvest COA, and then the material is processed, as it's processed, it's tumbled and THC is lost. So by the time it's in the package, that value will almost always be less than a harvest COA value. It needs to be tested just into the package or just before that process. The second thing is, in my first study, I looked at different sizes, and for the 1 gram packages, they were much below, some were 40% below label claim. However, when I did 5 different packages, and those were within sort of 5 or 6% of each other. So it was really actually quite tight because they had the smallest buds, included bud fragments. And these ones are typically 30% below the biggest buds. So there's an issue there. There's a size dependence. So the companies can produce, and as we've seen from our data, the variance between packages isn't high. The variance between labs is probably relatively constant. So you can kind of get an estimate. And it is actually very impressive with how tight some of the value is. They're simply not testing it in finished packages. And there's no consequence of having an artificially high value. Those 2 things add together for, of course, that's why we're getting it.
Thanks for that, Rob. Okay, we're getting into about an hour and a half territory here. So last chance for any questions here. Sorry, everyone. There's a lot of questions and comments in the Q&A, but we've kind of touched on a lot throughout the presentation in the last kind of 40 minutes of discussion. So panelists, I'll give you 1 more chance here to tackle anything, and if not, we can wrap it up. I do have 1 or 2 more questions for you myself.
There are a few questions regarding the sample size. Maybe I can lump them together. Personally, I think that everything less than 20 grams cannot be representative of a lot, and that's really pushing downward. The European Pharmacopoeia does have a very good sampling procedure based on your lot size. So it is true that 3.5 gram is probably not a full representation of the lot, but it's currently what's in package. And as Rob said, a smaller bud will have a higher variance than larger bud. So when we test, honestly, we always ask our customer to send us at least 20 gram. We won't use 20 gram. We will use 0.2 gram or something like that. But if we homogenize one bud, it's not representative of a lot, and there's no way that it's representative of a lot though. So I think that this is something that is quite important. And I know that some jurisdictions in the US have a standardized sampling method, and some also require the lab to do the sampling. I don't know if it helps. I don't know if it just create another point where you can prosecute yourself or whatever. Health Canada, back in the date of the hemp legislation, the sampler needed to be approved to do the sampling for the hemp. Maybe this could be reinstated. I don't know. I have no suggestion regarding this, but we are aware that sampling size does have an impact on the result. And I think that this is important for all QAP to be aware of. And there was another question in this regard is, I think it's, I think it's all right. So yeah, so that's one of my point.
Thanks, Hubert. All very important points. And you know, what it comes down to it is, I think, you know, in the sample to spreadsheet chain, you know, obtaining that representative sample is so important. And yeah, there is a lot of pressure from provinces, from clients, B2B, and medical consumers to get that high THC, because we know that's what sells, especially on the dried cannabis. But I think my hope is that the industry itself finds a way to get out of that cycle. And I think though the best way to break that cycle, what Rob and others have mentioned, is to enforce better regulatory and compliance measures on the sampling side, on the lab testing side, maybe mandate ISO accreditation or some sort of third-party validation or critical analysis of those methods. And you know, the provinces, what Ontario is doing, the OCS is doing, you know, it was interesting. We saw a lot of kind of pushback from the industry, because many probably on the analytical chemistry side said, this is great, this is what we want. And but others said, well, great, more variability and more pressure and more extended timelines. And you know, someone involved on the business side, I feel that as well. But we have to break this cycle. We owe it to the consumers, we owe it to this industry. That's the point of a regulated industry, to have a fair and level playing field. So, you know, this industry is maturing. We're about year 10 since the MMPR came into effect, and about 5 years into legalization. And these things take time. And I think the pressure that we as an association, as an industry, exert on the regulator, we see that they listen. It takes a little bit of time, but they're here on this webinar. They're out there knocking on doors. And I think we just have to keep doing this. So I guess as a final sign off, are there any plans here to do another study, expand this study, expand the number of labs, do this regularly? Has there been discussions on that, or where are we at with something like that?
So to be quite candid, I think we've developed a process to qualify labs. I welcome any other lab that wants to participate in these types of things to go into we'll send them PT material and real material, qualify them against the other labs. This could potentially take some pressure off QAPs, that they have a lab group. Of course, the ISO 17025 auditors will do that as well, but I know it takes time to get that accreditation, and it's an onerous project. But we do have a mechanism to help the labs, right? We are going to be producing more publications on this. And the interesting thing, the first time I did this on packaged dried flower, and the second time we did with this study, even though it's still more than half, before it was 80%. So it does seem to be improving. What measured matters. And if we look at another industry, let's imagine caffeine pills that you buy at London Drugs, instead of having 100 milligrams, came back at having 60, right? That problem would be solved immediately. London Drugs would give you your money back. They'd be holding the producer to account, and the Health Canada would make sure that problem was corrected. There would probably be a recall, right? And so those products, there's no problem. They're not They are medicinal type products. If you take something like caffeine, they don't have benefit. So we can do that. We can solve this. But if we don't solve it in cannabis, we're going to be embarrassed across the board. And so our international reputation gets hurt. I really hope Health Canada, I'm so grateful that we legalize. I'm so grateful that we're leading the world. Let's do the job properly. Simple as that.
Yeah. Wholeheartedly agree, Rob. We have an opportunity here in Canada to really lead, and we have, but you know, I think we can do a much better job, especially because it appears that the rest of the world is still lagging behind. I think all of us expected a bit more legalization throughout the world than we see today. Okay, final question. If anyone wants to contact any of you, has questions about the study, perhaps is interested in supporting future work or collaborating, who should they contact and how?
I think they could contact any of us at the moment. We're still in the process of discussing how we will approach the future. Like we will be keeping this free form. We will try to do something more crystallized to adjunct ourselves to another organism. So there's a discussion. This is still pretty early. I think that everyone here would love this to be a recurring experience. I think that, as Rob said, we are planning on publishing a guideline on how to do these procedure, these interlab testing with the market product. Because I do expect that the cannabis industry is not the only one that is affected by this, and I know for a certainty that it is not the only one. Rob is the main instigator, so I would say that it's pretty safe if you can contact him. But if you contact any other one, then we will make sure that it's forwarded to the right person and that everyone is kept in touch.
Yeah, I think LinkedIn is probably the best avenue to find some of you on there and then send a message and take it from there. So, okay, I think we're coming up to 90 minutes on behalf of our association. Thank you so much, all of you, Hubert, Rob, Andrea, and Thomas, and Denise, who couldn't be with us today, and all the labs that participated and all the people that provided funding. We were very happy to have the opportunity to provide some funding. We know how difficult this is. We know that you've all volunteered your time. And hopefully this is just the first of many of these discussions, and hopefully we can continue these discussions, maybe on webinars or even at our summit. So for those of you that are still around, there's still over 100 of you on here, but please check our website out, c45association.com. Please consider becoming a member, supporting work like this, and hopefully we see you out there in Hamilton on June 7th, c45summit.com. Tickets are $99, available today. And we're hoping to have some of us from the association and those on the call here represented, and should be a lot of fun. So thanks again, everyone, and stay safe out there, and we'll see you soon. Bye-bye.
Thank you.